So question 6, it says a reflex is a rapid response to a stimulus. The withdrawal of a hand when a finger touches a hot object is an example of a reflex. I'm sure you guys all had that experience, right? If you touch something really hot, your hand's like, woo, that was hot. Okay, so it says describe the pathway of a nerve impulse and the reflex arc involved in this response. So on my channel, under the 3-minute exam question series, there's a video called how to get full marks, reflex arc. See that? And then there's basically this whole process. Basically, you have to go through SR, SRM, you have to mention all of this. So let's start. So you start with S, the stimulus. The stimulus in this case is the hot object. The hot object. Then it's going to be detected by the R, which is the receptor. So detected by the receptor. What is the receptor, by the way? That's their skin, the skin on your finger. And then it's going to be picked up by the first neuron, which is the sensory neuron. And this then pass on what we call the electric impulse to the next neuron, which is the relay neuron. Relay neuron, which then continue to pass it on to the next, which is the motor neuron. And then finally, the motor neuron is connected to what we call the effector, which is basically the muscle, right? Which then contracts so that your arm would move away from the hot object, and therefore you would not be damaged. This is a very important survival mechanism to protect you from being burnt, basically. Now, there's one more point a lot of my students forget, so please don't be one of them, is remember, in between these neurons, how is the message being passed on? It is passed on, I'm going to put this little star up here because there's no space, is these things called synapses, right? So synapses. And in the synapses, there are neurotransmitters, which just continue to pass on that message so that it can be transmitted throughout the neurons. So all of these keywords, please ace the reflex arc question. Part B, the photograph shows an African land snail. When an African land snail is touched on its eye stalk, like doop, the snail goes back into its shell. The snail then remains in its shell for a period of time. A student investigates the effect that repeatedly touching the eye stalk has on the time the snail remains in its shell. Again, guys, just by reading, you should be able to figure out the independent and the dependent variable. It's always independent variables effect on dependent variable. Oops. Effect on dependent variable. So here it says the effect of touching the eye stalk, that's the independent variable, on the dependent variable, which is the time that it remains in the shell. Okay, so you have to be able to identify IV and DV from reading the question. Okay, but moving on, this is the student's method. So what he does is he gently touches the eye stalk of the snail with a cotton bud so that the snail goes back into its shell. Measures the time the snail remains in its shell, and then after the snail has come back out of its shell, touch the eye stalk again so that the snail goes back into its shell again. Measure the time the snail remains in its shell and repeat the stimulus 6 more times. That's a fun experiment. So that's what he's doing. And it says, the student's results are shown in the table below. Plot a graph to show the effect of the stimulus number on the time the snail remains in its shell. Join your plots with ruled straight lines. So when you plot graphs, remember, you need to have the one on the left here, the stimulus, that is the X, and this is the Y. So you got to be plotting it correctly, right? So they've given us this graph paper down here, and we're going to plot this together just to see how good our graph plotting skills are. So your first thing is to get the axes correct. So remember, X is this column here, so the stimulus number goes on the x-axis and then the time goes on the y-axis. Now if you just put your mind in the examiner's shoes, this is what they are looking for when they are marking your graph. They're looking for SLAAP. SLAAP. S stands for the scale. So we're going to determine the scale. Scale meaning like, is it going to be 1, 2, 3, 4, 5 or 2, 4, 6, 8, 10 and so on, scale. And then is the line, whether it's a straight line or a curve, depends on the question. And then A is the axes. These both A's are axes, okay? The first A is the orientation. In other words, making sure you get your X and the Y in the correct places. In other words, you cannot put time here. That would be wrong, right? And you know the correct order of the axes by looking at the table. The first column on the left is always the X, and then this one is the Y. So that's the first A. The second A axis, that A stands for labeled. Labeled with units, right? You need to have units. And then finally, P are the points, which you're going to plot very, very correctly. Okay, so let's do this plotting. Now, the first one is scale. Stimulus number. So stimulus numbers goes from 1 to 8. So I'm going to try to remember, when you do scale, you want to spread out your points as much as you can across the graph. Don't use like a little bit of the graph. And obviously you need to have all of the number on it. So if I put obviously zeros here, if I go up in 1, 2, 3, 4, you can see how this doesn't work because I need to go all the way up to 8 and 4 doesn't work, right? So that's how you figure out the scale, by just looking at it and using some common sense. So we got to go up maybe by 2s now, 2, 4, 6, 8, and that is perfect because I need to go up to 8. Okay, and then for the y-axis, how are you going to go up the scale? Are you going to go 1, 2, 3, 4, 5? That doesn't work because I need to go all the way up to 12, right? So what about 2, 4, 6, 8, 10, 12? Ah, that's perfect. The other one would have been something like 5, 10, 15, but that would be dumb, right? Because if you do 5 here, 10, and then 15, then you just end up using just this bit of the graph. And that's not using most of the graph, and you won't get most of the marks. Get it? So you want to use as much of the scale and also use a reasonable scale. So this is going to be 2, 4, 6, 8, 10, 12, and 14. And then we're going to go ahead and plot this. Okay, so I've plotted the graph right there, and I've joined it with straight lines. Okay, now below here, the next question asks, describe the effect of increasing stimulus number on the time the snail remains in its shell. Okay, now when you're describing the graph, you want to discuss any trend. Trend means, is it positive, meaning the graph is like this, or is it an inverse relationship, which is usually like that? So the trend, okay? And then you also want to describe any changes, any gradient, so any major changes. So having that as a guide, let's try to answer this. So is it a positive or inverse graph? It's an inverse graph because it is going down, right? It's going down. So you would say this as the stimulus number increases, time decreases. That's how you describe it. So you would say, need more space down here. You would say it's this one, right? So you would say as stimulus number increases, time, be specific, time in the shell, right? Time in shell decreases. So that is the trend. And then you also don't stop there. The saddest thing is when people just kind of stop there and then move on and you don't get the second mark. This is a 2-mark question. The second mark comes from any changes in this graph. So do you notice when it starts to kind of really have a change? At what stimulus number on the x-axis? If you observe any time before, roughly where? Any time before 5, which is here, right? It's going kind of flat, down, up, down. It's kind of like sort of wavering, but mostly flat-ish, not much difference. And then anytime after 5 number of stimulus, it plummets. Get it? So you want to identify this change. I'm going to highlight this point here just so that you really kind of get the point I'm trying to make with these graphs, is that you look at the changing point, the turning point. So that's exactly what you would say. You would say, for the changes bit, you would say before 5, right? Before 5, remains similar. The time remains similar. And then after 5, the time drops a lot. And you need to express that. So steep drop, steep decrease in time. Steep decrease in time. So let's just understand what this means. It means that when you poke the snail 1 time, 2 times, it stays inside the shell for quite a long time, like up to 12. That reminds me one thing. What am I missing in this graph? Can anyone tell me what mark am I missing? I got the scale, right? I've drawn the line. I've plotted it beautifully. I've got the axes. So I've got 4 marks. What's the missing mark? It's the labeling. So I need to label the unit. Don't forget the unit. So time in shell. Time, is it in seconds? Or is it in hours or days or light years, right? Time in this case is in seconds. See that? It's in seconds. You have to say that. You have to have that in your graph. So always check. Okay, so basically it means that when I am poking the snail, anything less than about 5 times, it stays in the shell for quite a long time. It hides in there for up to 10 seconds. See that? But if I poke it again more than 5 times, like it actually doesn't stay in the shell very long. It just kind of comes back out. It just stays really short. Like after you poke it for 7 times, it just stays in the shell for about 4 seconds, 5 seconds, and then it comes straight back out again. So try to picture what's going on, because that is what we need to answer in the last part of the question, which says suggested benefit to the snail of the difference in response to the repeated stimulus. In other words, what is the difference? Do you guys know? The difference is the change that you've identified, this before, 5, and after, right? Why does it have less and less time remaining inside the shell after you've poked it many times? And that's kind of common sense, right? Imagine if you're the snail. If someone pokes you, you might think it's real danger. So you would shrink back into your shell and hide there. But if the same thing pokes you again and again and again, you're starting to think, come on, man, is this real? So you don't really register this as danger anymore, so you just stay in for a little while and you come straight back out again, because it's important to come back out because you have things to do, right? You don't just hide in your snail in your shell all day. So let's try to express this in words. So same thing. So the before 5, so this bit here, when it's less than 5 stimuli, right? So it means that, think about it. When you're first getting poked just a few times, you don't really know if it's danger or not. So you are worried that it might be something dangerous as a shell, as a snail, right? So concerned with danger. All right. So prevent getting eaten. This is a biology paper. So you want to think about those keywords like predator, getting eaten by predator. So you want to stay, the shell helps to survive, helps the snail to survive. Okay. And then you want to explain the afterwards. Anything more than 5 stimulus, more than 5 stimuli, why does it have a different response? It doesn't stay very long at all, right? It stays a much shorter time after five pokes. And why is that? Because think about it, it's just the fact that the snail no longer considers the stimuli or the stimulus as danger, right? Or in other words, it kind of recognizes it is not dangerous, right? So it needs to come out sooner. For what? For food, okay, or for movement or whatever it was doing before you decided to poke it so many times for your experiment. Okay? So question 7 says coronary heart disease is caused by damage to the walls of the coronary artery. Explain 2 differences between the structure of the wall of an artery and the wall of a vein. Now before we go further, I just want to show you guys what the coronary arteries actually are. They're not the arteries that you guys think of sometimes, which is the aorta, the renal artery, the hepatic artery, even the pulmonary artery. Those are really big, guys. Those are like thicker than maybe the width of 2 of your fingers, okay? But the coronary arteries, they're actually incredibly thin. They're actually these ones on this diagram. So you see how you might recognize this diagram, right? This is the heart. This one, that is the aorta. That's the aorta. This is the pulmonary veins, and then this is the pulmonary artery. Okay, so just a quick revision there. So you see these big blood vessels. Look how thick the diameter are for these. Look at them, right? That's very thick. Now, the coronary arteries, on the other hand, they're these really thin ones that kind of just supply the heart itself. I repeat that again. The coronary arteries carry oxygenated blood to the heart itself, not away from the heart, not going to other organs, to the heart muscle itself, because the heart muscle itself needs that oxygenated blood in order to pump. Okay? Now, the other important thing about this is that they're really thin. Look how thin they are compared to how thick the aorta is, right? The danger of that, why does it cause this disease called coronary heart disease, which is actually very common? The danger is that since they're so thin, they're easy to be clogged up. By what? By blood clots or cholesterol, lipids, all the unhealthy stuff from your diet. They're easy to be blocked. And if they're blocked, then you cannot supply oxygenated blood to the heart, and then that person may have something called a heart attack, which is quite a common cause of death. Okay, so that is basically what a coronary artery is. So just next time you eat something unhealthy, remember you're clogging up those very thin coronary arteries, and you don't want them to get clogged, because then your heart will get a heart attack. So anyway, moving on to actual questions. So we're talking about the differences between the structure of the wall of an artery and the wall of a vein. So let's bring up my beautiful drawing here. This is an artery, this is a vein. So let's compare the differences. So let's be very structured. There are really 3 differences between arteries and veins. The first one is the amount of muscle. More muscle in the artery, less muscle in the vein. The second thing is the elastic fibers. Again, more in the artery, less in the vein. The third one is the valve. No valves in the arteries, yes valves in the veins. So when you have that kind of sort of organization in your mind, then you can start to talk about it. So the structure, so let's write this, the structure. So what is actually the difference in the structure, and then explain, right? So never just write it down, you have to explain. That's the reason. Understand? So the structure is that for the artery, for this bit, the artery, we have thicker muscles, and the second difference is more elastic fibers, and the third difference is no valves. So once you write down these, then you write down the corresponding reason. So muscles are there to control the blood flow, right? Control blood flow, because when they contract, then the blood can be squeezed along. The elastic fibers are there for a specific reason, and that is to withstand the high blood pressure in the arteries. Remember, we're talking about the arteries here, right? In the arteries. So do you guys understand? Try to picture it. When the blood pressure is really high in the arteries, it requires that elasticity in the wall of the artery to be able to deal with that high blood pressure. Otherwise, the whole thing would just collapse. Get it? So that's why you need a lot of elastic fibers. And then finally, no valves. Now, what's the reasons of not having valves? Think about it. Valves are there to prevent backflow of blood. Would there be any backflow of blood in an artery? Would you expect there to be? No, because blood only flows backwards when things start to get really slow, and they're supposed to be going forwards, and then they start to decide to fall back. That doesn't happen in an artery, because an artery has a high blood flow speed. That's high blood flow speed, and therefore there's no backflow of blood. So there's no valves required, okay? But you can still mention the function of valves, which is that prevent backflow of blood. However, you say that the, but blood pressure is sufficient. So no valves needed. Get it? Now, you guys can also, just take note, guys, this is a 4-mark question. So where do the marks come from? It comes from 1 point about the structure and then 1 point about the reason. So this is 2 marks. And then another 2 marks on 1 point on the structure, 1 point on the reasons. You get it? So if you just wrote this, this, and this, you're not getting more than 2 marks because half the marks come from the green, the reason. Understand the exam skill right now? So structure and then reason. Now you could also have written this the other way around for the veins instead. So you could have said thinner muscles, less elastic fibers, and yes, valves because of the reasons correspondingly. Get it? So be very structured in your answer approach. Okay. Part 2 says, the diagram shows a cross-section, means they basically cut through the blood vessel of a coronary artery from a healthy person and from a person with coronary heart disease. So this is a healthy person, which is you right now, hopefully, and this is a person with coronary heart disease. Notice that all these stuff is clogged up, right? That's not good. That is not good. So this bit here is the lumen. So you see how the lumen here is much smaller. So basically there's less space, room, volume for the blood to flow. And that's a problem because the blood is flowing to very important places, right? Okay, so the heart needs to respond to extra demands during exercise for the person with coronary heart disease. So it says here, explain why during exercise, think about what your heart is like during exercise. It's already pumping faster. Why? Because you need to pump more oxygenated blood to your muscles. Okay? The heart of a person with coronary heart disease would not respond as efficiently as the heart of a healthy person. So I think this is quite obvious, right? So you would just talk about what you say. So here you see that you would say that the lumen is smaller because more blockage, right? More blockage from what? From, if you don't know what it is, you don't have to write it, but usually it's cholesterol and lipids and stuff like that. Cholesterol. That's why your diet, a healthy diet, is very important. So lumen is smaller because blockage. Now, how does that make it worse for the person doing exercise? How does that make it worse for the person doing exercise? If the lumen is smaller, that would mean there is less oxygenated blood delivered, right? Delivered to what? To what? You're doing exercise, right? So where should the blood be going? To your brain? Not to your brain. You don't need to think to do exercise. You need muscles to do exercise. So less oxygenated blood delivered to muscles. Muscles. That's very suspiciously bad spelling. To muscles, right? And then don't stop there. When you think of oxygen, what does that mean? That means there's less aerobic respiration because that's what oxygen is for. It is for aerobic respiration. Remember the equation? Okay? And if there's less aerobic respiration, tell me what does that produce less of? What is the products of aerobic respiration? Oxygen plus glucose, maize, carbon dioxide, water, and ATP, right? So therefore, less ATP. Okay? So that is basically the main points for this answer. Part B, genetics and smoking are 2 risk factors for CHD, which stands for coronary heart disease. The alleles of some genes increase the risk of developing coronary heart disease in people. These are known as risk factor alleles. Remember, guys, alleles are just different versions of a gene. So, for example, if we have the gene for eye color, the alleles would be blue color, green color, brown color, and so on. Okay, so it's just different versions of a gene. So it's your genetics. Okay, so scientists compared the relative risk of developing coronary heart disease in people with different numbers of these risk factor alleles. The scientists also compared the risk of developing coronary heart disease for non-smokers and smokers. The graph shows the scientists' results. Now, if you just kind of have a think about what's going on here, whenever you read experiments, it's always interesting to know the IV, which is the independent variable, and the DV, which is the dependent variable. So what is the IV here, guys? What are they changing? They are changing, usually the IV is on the x-axis, so they're changing, or they're looking at the the impact of different numbers of risk factor alleles. So that is the alleles, basically, the numbers of risky alleles. But there's also another independent variable. They're also changing what else? What else are they changing? They're also changing whether or not they are smokers or non-smokers, right? Because they're looking at whether or not smoking affects the risk of coronary heart disease. So smoking is another independent variable. So this graph actually has two independent variables. And what is the dependent variable? That is what they're measuring, and that is usually on the y-axis. So that is the risk of coronary heart disease. Start to make sense of these graphs, okay? So that's the results. Okay, and it says give one risk factor for coronary heart disease other than genetics and smoking. Now, you've been students of mine for long enough. I want you to be have a systematic approach to talking about risk factors. Remember, for risk factors, there's always the ones that you can control, and then the ones that you cannot control. And that's important because once you know this, then you can change, you can improve the things that you can control. And what does this mean? So basically, look at what they already said. Other than genetics and smoking. So which one is something you cannot control? Can you control your genetics? No, you cannot control that, right? So therefore, genetics is something that you cannot control. So this is genetics. Whereas smoking, you can definitely control that. That's lifestyle, smoking. So this is, put in simple terms, this is basically your genetics, what you're born with, basically. That is that. And then the things you can control, we usually put that under the umbrella of lifestyle. It means that you can control how you choose to live your life. Get it? So smoking, you can control. What else can you control that will lower your risk for coronary heart disease? What else can you do? What else do you do in your life that's risky in developing coronary heart disease? Think about your lifestyle, diet, right? And don't just write diet, guys. Please don't just write diet, because what about the diet, right? High cholesterol diet is not a good thing. So this is another risk factor, high cholesterol diet. What else? Stress. So please don't be stressed. It's not good for your heart and your blood vessels. What else? Diabetes is a very common one. That's not that uncommon as a disease. Is when your body is unable to regulate the glucose in the blood. So diabetes also raised the risk of coronary heart disease, and also high blood pressure. So this is not just for your biology exam, is for your life, okay? So keep healthy and think about these lifestyle factors that you have control over and do something about it. Now, for the genetic side, apart from genetics, what else can you not control? Can you control your age? No, you can't. So the older you are, the higher the risk, right? So you can't do anything about that. Sex is something you cannot control. If you're male, you're female. Usually men have higher risk of coronary heart disease. So sex is something that you cannot control. Okay, so have a systematic approach. So I think we can get way more than 1 mark here, right? But you got everything, right? So part 2 of this question, it says comment on the relationship between the number of risk factor alleles and smoking on the risk of developing coronary heart disease. Now I've put the graph above here so you can still see it. It says use the data from the graph to help your answer. Now, this is 5 marks. When you see this on the exam, you know that you can't just go in randomly. You have to have a more systematic approach so that you don't miss anything, right? And that's why just now when we read the question, I encouraged you guys to think about the independent variable and the dependent variable, because we're discussing their relationship here. Okay, now I've had a look at this question, and here's my suggestion for the approach, because how can you make sure you have enough points to get 5, like all the 5 marks, right? So I've actually put down here these 6 areas that I intend to cover, and therefore it will guarantee full marks. So I'm going to talk about the overall trend of this graph. So let's just do these one by one, okay? So what is the overall trend? Overall trend means it is a positive correlation, right? So you just literally write that, positive correlation. And then, to say what you mean by that, it means that more risky alleles, cannot spell, risky alleles, make this bigger. Okay, so more risky alleles result in higher risk of coronary heart disease. And then also, remember, you're also not just looking at the alleles, which is here, few, medium, high, but also the smoker, non-smoker, right? So that is the second point already. So if you're a smoker versus non-smoker, who has a higher risk? So look at the colors, right? The smokers, which is a light color, this and this and this, they consistently have higher risk than the corresponding non-smokers. So you would want to write that. So you say that smokers have higher risk than non-smokers. Okay, for each allele group. Get it? In other words, you can you be more detailed. You can say for few risky alleles, the smokers have higher risk than non-smokers. For medium risky alleles, the smokers have higher risk than non-smokers. And for high number of risky alleles, the smokers still have higher risk than non-smokers. So smoking is clearly very, very bad for coronary heart disease. Okay, and then the third point is highest versus lowest risk. So this is who's got the very highest risk. So once you have this overall approach, then you can't really miss it, right? So the highest risk is here and the lowest risk is there. So you literally just write it. So you say the highest risk is the smoker plus risk high allele. Get it? Okay, smoker plus high. Yes, and then the lowest risk is non-smoker plus low allele. So that's number 3. And the fourth point is data manipulation. So they like to see you do a bit of data manipulation. Manipulation in a biology paper isn't a bad word, the word manipulation. It simply means they want to see some calculation. So for example, look at the few ones. For the few ones, if you are a non-smoker, your risk is 1. Non-smoker, right? But if you are a smoker, your risk is 2. So in other words, the risk of developing coronary heart disease in a smoker is twice as much, because 2 is twice as much as 1. That's what they mean by data manipulation. And this is the most obvious one, isn't it? Because it's literally 2 and 1. So you literally say that. So for few alleles, so that group there, smoker risk of coronary heart disease, it's double, because 2 is double of 1, right? Double of non-smoker. Understand? So some sort of data where you time something divided by something and so on. Okay, now the next point is very nice, is why? Because a lot of people forget this. They just kind of like comment on the data like, oh, this is high, this is low, positive or inverse relationship. But after all, this is biology, and they spent 2 years IGCSE, your whole syllabus, your GCSE, teaching you about the risks of smoking and all that. They want to see that you know this knowledge. So why does smoking have such a bad impact on coronary heart disease? You just have to start thinking about what is so harmful in the cigarette. What are the 2 things that are really bad in the cigarette? One is the carbon monoxide, right? And that's definitely bad for the blood vessels. What is another thing? Let's write this carbon monoxide first, okay? So the carbon monoxide causes coronary heart disease. And if you're not sure, just at least write down carbon monoxide, get it? Because that's already a keyword. Also, it causes plaques. Smoking causes plaque, or cholesterol buildup. In where? In the coronary arteries. Remember, they're really thin arteries, right? That get clogged up easily. So in the coronary arteries, and that's not a good thing. And then also, cigarettes have nicotine. Nicotine is very addictive, so that just makes it a very vicious cycle. It's bad for you, but you still crave it. That's very bad. So don't smoke. Do not smoke. Okay, next point is limitations. People forget this. It's like a mark you will lose, but if you know to talk about it, it's a free mark you'll get. So what does limitations mean? Is you have to find or discuss what about this data is not good enough, basically. So limitations is usually referring to the control variable. So you think about what variables have not been controlled, or at least not mentioned, because if they didn't mention it, you don't know if they control for it or not, right? So what would you expect the people here, the experimenters, to control? What should they be keeping the same? They obviously got a bunch of people and looked at their genes, and they looked at the risk of coronary heart disease. But what do they not mention? What should these people have the same of? They should have the same age, isn't it? Because if you're older, then your risk will be higher. Even if you are or are not a smoker, it doesn't matter. Your age is already affecting the results. So they should have the same age. You don't know. Do they have the same age? What about the sex? And that's actually, guys, why the last question encouraged you to think about what are some of the other risk factors, because this is supposed to guide you for the next question, which is telling you to comment on this experiment. So they didn't actually think about the age or the sex or whether or not they have all these conditions, right? Do they have stress in their lives? Do they have diabetes? Because it would also affect the dependent variable, which is the risk of coronary heart disease. So just mentioning a few control variables already gets you that mark. And here's a final cherry on top, is just mention the data, what we call the data sample size. So we don't know how big is the data, as in like how many people did they use. If this experiment just used 10 people, like 5 smokers and 5 non-smokers, are you going to take this experimental result seriously? Probably not, right? Because there's only 10 people. It's not a very big experiment. So the sample size is talking about how big is the number of people used in the experiment, because that actually reflects how valid the answer is, how reliable the results are. So always talk about control variables and the sample size for the limitations point. So once I've sort of color-coded everything, I hope I give you an approach for talking about graph and data interpretation in biology. These kind of questions do come up, and they're usually lots of marks, and have a systematic approach, such as the color approach I have here, so that you don't miss any points. And some points are very easy, like limitations, data manipulation. You can easily write something about those, okay? So question 8, lactose is the sugar found in milk. So let's say lactose looks like this, okay? And then the lactose needs to be digested in humans by an enzyme called lactase. So this is the lactase enzyme. So the products can be absorbed, because if it's too big, you cannot absorb that into your intestines, right? Okay, so all human babies produce lactase in their intestines. However, people who are lactose intolerant stop producing lactase and are unable to digest lactose sugar as adults. In other words, if you're lactose intolerant, you do not produce this enzyme anymore. So lactose intolerance is a genetic condition caused by a recessive allele, small d. The ability to digest lactose is caused by a dominant allele, capital D. And then it shows you a family pedigree diagram. So before I go any further, I have a whole video on pedigree diagrams and decoding it. There is almost always guaranteed a question on this, because genetics is such an important part of biology. There's almost a guaranteed chance this will come up on the exam, and I want you to be able to do the pedigree exam perfectly. So this is the video on my website. It's called Everything You Need to Know About Genetics and Pedigree Diagrams. So find that in the topic video playlist, so that you could sort of I go through several pedigree diagram exam papers in there, so you have a taste of different types. Okay, so let's come back to this question here. Now, a few more keywords we need to revise, which is phenotype and genotype, right? So remember, the phenotype is what you actually see in the person, which is whether or not they're lactose intolerant or not, okay? So that's the phenotype. And then you have the word genotype, and then genotype is basically the genetics that you have. Now remember, for every gene you have, in this case the lactase gene, which digests the lactose. So the gene for lactase, how many copies do you have in each of your cells in your DNA? You have 2 copies, because you came from a mother and a father, and they would have given you 1 copy each. So you will have 2 copies of the lactase gene. So your genotype is basically 2 letters, big D, or big D, small d, and so on, okay? So once we know some of these terms now, we can start to think about this. Now, they've already told you that this disease, this lactose intolerance disease, is a recessive disease. In other words, if you have the disease, you will only have the small d, because that's what recessive means. Dominant means 1 copy of the gene, you will see the disease. Recessive means you need 2 copies of the gene to see the disease. So since this is a recessive disease, you need 2 small d's. And therefore, those that are black here, because guys, we need to, what are we doing in this question? They're asking you to basically draw the genetic diagram, which is the Punnett square, the 2 by 2, for number 4 and 5. So in order to do this, you have to figure out the genes of 4 and 5. And in order to do that, you have to figure out the parents, because otherwise how can you know number 4, right? So we have to basically figure out the genotype of everybody in this diagram. Okay, so let's go ahead and do that. Since we know that this is a recessive condition and that the black color is the ones that is lactose intolerant, right? With lactose intolerant. So the black ones are the ones with the disease. So we already know that the genotype for the black ones is small d, small d, small d, small d, small d, small d. Wasn't that easy? Okay, now we need to figure out the white one. Now the white one, think about this white one. White means you do not have lactose intolerance. That means you must have a big D, which is the normal gene that produces the lactase so that you can digest the lactose. Now, this white, to be white, this white circle, it could either be big D or big D small d, right? Because both of these cases, you are able to digest lactose because you have the big D. So which one is it then for number 2? Does number 2 have big D small d or big D big D? You have to think down towards their kids. See, they had 2 kids. They had a girl, circle is girl, and they had a boy, which is a square. And since the girl, look carefully, the girl has small d, it means that she must have gotten 1 small d from her father. And where did the other small d come from? Did it also come from the father? No, right? No. She would have gotten 1 small d from her father, and then the other small d must have come from the mother. And therefore, number 2, the mother must have a small d. Do you understand this detective game? I really love this. So this mother must have a small d. But since she's white, means that she doesn't have the disease, she must also have a big D. Therefore, the number 2 here is big D, small d. So now let's figure out number 4. Number 4, remember, got 1 copy each from their parents, right? So what copy did number 4 get from his mother? The big D or the small d? Remember, number 4 does not have the disease. So he must have gotten the big D from his mother so that he doesn't have the disease. And then he obviously got a small d from his father because that's the only thing that his father has to offer him, it's a small d. So this 4 would be a big D, small d. And then you just check to make sure everything makes sense, right? That the white ones have a big D and the black ones all have small d's. Once we have that, now we can proceed to draw this genetic diagram, which please have the following approach. You need to, first of all, show the parents. Remember, this is 4 and 5. So this is the father, right? This is the father. And then this is the mother, father and mother. So for the father, and then the mother, because I don't want you to lose any marks for this. You need to have the following information. You need to have their genotypes. So what is the father's genotype? We just spent ages figuring that out, right? The father's genotype is capital D, small d. So that's what you're going to write, capital D, small d for the father, big D, small d. And what's the mother's genotype? The mother's is small d, small d. Okay, so that is the genotype. And then after you have the genotype, you must also write the gametes. What the gametes mean, guys? What the gametes mean is like what type of alleles do they have to offer? So look at the father. He can offer both big D and small d. Get it? So he's able to offer both the big D and the small d. Whereas the mother, she only has a small d to offer. So that's what gametes mean. Remember, gametes is basically the sperm and the egg, right? Because they join together in order to make the zygote, which is the first cell in the offspring. So now we can go ahead and draw the genetic diagram. So let's do that. So for the father, so let's have the father on this side, father, and then like this, and then the mother, and then you would list out their their genotypes, right? So for the father, it's going to be big D and small d. And then for the mother, it will be small d and small d. And once you have that, you can now go ahead and fill out the rest of the table, while I'm going to go ahead and make this table look a little neater with very straight lines, because it's nice to have very neat tables like that. Okay, so once you have that, then you can fill out the content of the table. So let's do that. So we have in the offspring, so the offspring is going to be in purple here. So this, you know how to do this, right? So this one will be big D, small d, this one will be small d, and so on, right? So let's do this lightning speed: big D, small d, small d, big D, small d, small d, small d. Okay, now remember the purple color here, that is the genotype. That is a genotype. And it's asking for the probability of the child having lactose intolerance, having the disease. Having the disease is not the same as having the genotype. It depends on what the condition is. So having the disease, which of these four has the disease? Which one? Tell me. Lactose intolerance. You need to, it's a recessive condition. So therefore, two small d's have the condition. So in this case, 50%. Understand? 50% is the chance of having the disease. 50%. Now, because the pedigree questions are very common on the paper, I want to just highlight again how you're getting these 4 marks, okay? How you're getting these 4 marks. You're getting 1 mark for writing down the genotypes of the father and the mother, which you get from figuring out the pedigree diagram, okay? So write down the genotype like this. That's 1 mark. The second mark comes from writing down the gametes of the father and the mother. The third mark comes from getting these 4 little squares correct, which is the genotype of the offspring. That's the third mark, number 3. And then the last mark is obviously getting the probability correct. So that's how you get the 4 marks. So don't miss 1 and 2. A lot of my students, they forget 1 and 2. They just go ahead and draw the table, number 3, and they get 3 and 4. So you really should be having 1 and 2 clearly stated in your answer, the genotype and the gametes of the father and the mother. So the next question is very interesting. It says, people with lactose intolerance often get diarrhea if they drink milk. Suggest why people who cannot digest lactose get diarrhea if they drink milk. Basically, to explain why lactose intolerant people get diarrhea. Now, I want you to imagine you're going to zoom in to someone's gut. Okay, so this is someone's gut. And you know, in the gut, there's these little things called villi, right? That's the villi and there's the villi. Like that. Get it? Now, the lactose is the thing from the milk, remember? So let's say it's that little. So these are the lactose that you have just drank because you had some milk. Okay, now normally what would happen is that these should be broken down by lactase so that it becomes smaller and therefore small enough to be absorbed into the villi. Remember, the villi have high surface area and a lot of capillaries, and therefore the nutrients get transported into the blood and go to the rest of your body. That's the whole point of digestion and absorption, right? So that's what happens in a normal person's body when they have the lactase enzyme to do that job. Get it? However, in people that cannot, these are lactose intolerant people, they do not have this enzyme. And therefore, where do these red little triangles stay? Where do the lactose stay? They stay inside the gut. They stay inside the gut. And of course, as you go down the gut, this is the small intestines. By the time you get to the large intestines, that's where poop is made, right? So this is inside the gut, and they never ever leave the gut because they cannot be digested to be small enough to be absorbed into the bloodstream. Now, why does that cause diarrhea? Is because, think about it, when these lactose stays in the gut, some of the water molecules is going to end up also going into the gut, because if they get absorbed into, imagine, if they do go into the villi, then the water will also follow, because water goes from high to low water potential. Now, since they're not getting absorbed, then not so much water is going to get absorbed either. Make sense? I'm going to repeat that again. Since these red triangles, these lactose, are not getting absorbed into the gut, water is also not going to get absorbed into the gut. And since water is not going to get absorbed into the gut, what is it when your gut produces watery poop? That would be diarrhea. Okay, so therefore how would you explain this in words? You would say lactose, let's write neater, lactose not absorbed. And therefore, water also not absorbed. It's not really good to write this because of course some water is still absorbed, right? So I think we should make this better. We should say less water is absorbed. Less water is absorbed as a result. And give me more details, right? Lactose is not absorbed into intestines, right? Into the intestines. Is not absorbed into the intestines, and therefore water is also not absorbed into into there. And you can talk about the water potential. So if you're not really sure how, you can just be a bit more vague. You can just say the lactose affects osmosis, right? Just at least talk about osmosis because you talked about water movement, right? So affects osmosis and the water potential. And there's also a completely different point, is that when the lactose stays in the gut, it also it can breed bacteria. And the bacteria would also make you sick and therefore get diarrhea. Okay, because the bacteria will break down the lactose instead and end up giving you very gassy feelings that can result in diarrhea. So next time you know someone that has lactose intolerance, you can explain the whole process to them and they'll be very impressed. Okay, the next one is obviously a much more cuter question because I did some drawings for you guys. So here it says, milk is a nutritious substance that provides several food groups. In areas of the world where milk is a main part of the diet, fewer people are lactose intolerant. Isn't that interesting? So explain how natural selection has resulted in fewer people being lactose intolerant in areas of the world where milk is a main part of the diet throughout their lives. So I'm trying to make this sort of a bit more visual. So the parts of the world, areas of the world, okay, let's make this here. Areas of the world where milk is a main part of the diet, by those areas, we're talking about this area on the left hand side. Okay, so this is a country where they love milk, and their milk is used a lot in their diet. I'm sure you guys can think of some countries where milk is always something that you have every day. Okay, so that's this part of the world. Whereas the right hand side, this country is somewhere where they just don't have a lot of milk. Now, before we proceed further, I hope you guys know the main points about natural selection. Automatically, you guys should mention the following, right? Natural selection, what are the main words? Put a star there, put a star down here. Even without the milk example, you need to have the following keywords for any question on natural selection, which is mutation. You can talk about random mutation occurs. The next point is what? That causes variation in a population because there's a mutation, so therefore the genetics is slightly different between different individuals in the population. So variation. And then so what some will have a survival advantage, and in this case the survival advantage is the ability to do what? Ability to digest lactose. Because we're talking about the advantage in the blue country, right? Ability to digest the lactose. And why is that a survival advantage? That means you're not losing all the nutrients through diarrhea and you're actually growing healthily, right? So that's a survival advantage. And then don't forget, finally, they're going to survive, reproduce, and pass on the gene. And in this case, it's the lactase gene. So let's try to represent this sort of diagrammatically, right? So on the left country, the blue country, let's say a mutation let's say all of these people right now don't have the lactase gene, and then you have a mutation where you have, you know, some of them do have the lactase gene, so these two have it, and the other two don't, right? And therefore, who would have a survival advantage in a country that has lots of milk products? These two are going to have a more chance of living because they're eating better, they're growing better, they're not losing all their nutrients, they're healthier, right? So therefore, there's more chance that they would go on to reproduce. And therefore, the next generation of kids, because they came from parents that have the gene, right? And then, of course, they could produce more kids and so on and so forth. All of them will at least have this lactase gene. Get it? So therefore, over time, there's going to be more people with this gene. And this is the beauty of natural selection. It naturally selected for the lactase gene so that the people can grow healthier because they're able to deal with the milk in their country. Whereas in a country where they don't really have milk, it doesn't really matter who has this mutation or not, who has it or not. Even if you do have it, it doesn't give them an advantage, right? So even if these people have it, you know, at the end of the day, they could have kids. So the products were the offspring, yeah, it would also, some of them have it and some of them don't. So you see how here we went from 50% having the gene to 100% having the green gene, whereas here it goes from 50% to still 50% because there's no advantage in having the gene. So it just kind of stays constant. That, you know, the frequency, what we call the frequency of the gene stays constant in the population. And this is how selection pressure acts. The selection pressure for this is the milk that they're having. Part B of question 8, milk can be used to produce yogurt. The diagram shows a fermenter that can be used to produce yogurt in the industry. So you have this fermenter. They love to ask about fermenters in recent years, okay? So please know how a fermenter works. So here's a fermenter and it's making some yogurt. And the first part says, explain the role of the stirrer in the fermenter. So even if you have no idea, I think you can kind of figure out what a stirrer does just from common sense, right? It mixes the contents, right? And it's, you know, it helps us spread out the nutrients, you know, even consistency, get it? So it's not really thick in some places and really thin in other places, so you need it to be very even throughout. This is 2 marks though, so I want to talk about something other than mixing. What else is going on here? Well, you know that if you're making yogurt, there's going to be bacteria involved, right? And if bacteria is involved, the temperature matters because bacteria will die if it's too hot. So having the stirrer, you guys should know this from your experiments where you have stuff in a beaker and you heat it up and you usually have a stirrer, right? So what does stirring does? It helps to spread out the hot water so it's all even. Get it? So it's to maintain, the stirrer in this case, apart from just mixing, it maintains an even temperature throughout. In other words, it's not like really hot in one place and really, you know, not as hot in the other place. It keeps everything really balanced. And the next part, it asks, explain the role of the water jacket in the fermenter. So the water jacket is like this thing. It's basically a jacket, so it kind of wraps around this whole fermenter. You see that? So what is the point of the water jacket? The whole point is it helps to cool things down. So if it gets really hot in here, the water that gets run through here is cooler water. And if it's cooler water, guys, I hope you understand what's going on here. It's a water jacket. They're not pumping water into the fermenter, okay? They're just wrapping the fermenter in a jacket, and then they can put water into the jacket. And therefore, it cools down the contents inside the fermenter. They're not diluting it with water, okay? So therefore, what does the water jacket do? It helps to cool the fermenter, cools the fermenter. And notice it says explains. You have to say why. Why do I care about cooling the fermenter? Because the temperature matters. Why does the temperature matter? Because we have bacteria. What kind of temperature is good for bacteria? Optimum temperature, right? Optimum temperature to maintain, to keep optimum temperature. And why is that? Don't stop there. Tell me more. Why do we have to keep the optimum temperature? Is to prevent what? To prevent the enzymes from denaturing. So all of this is like all linked up, isn't it? The temperature, the optimum temperature, and then prevention of denaturing. So that is a fermenter. So question 9 says, transgenic varieties of tomato plants have been produced that can photosynthesize more efficiently than natural varieties. To make the transgenic tomato plants, a gene is inserted into a vector that is then placed into tomato plant cells. Describe how a recombinant vector containing a gene can be produced. So let's try to visualize what is happening here. So the gene that is able to photosynthesize more efficiently, that is going to be this little bit here. That is the gene. That's literally the gene that you're looking at. And then the vector is how you're going to carry that gene. And that would be a plasmid, okay? So this is called a plasmid. Now, what happens at the beginning is that this plasmid would have been just like that. It would have been a complete circle. And if you guys remember, plasmids come from bacteria. So this is a plasmid in the bacteria. But I need to insert this green gene into the red plasmid. In order to do that, I first need to cut. You see that? Cut at 2 locations in this plasmid. And that requires an enzyme. Can somebody tell me the name of this enzyme that does the cutting? That's a pair of scissors right there. This is restriction enzyme. So I'm going to use restriction enzyme to cut it so that therefore this little bit here, this section gets cut out, and therefore I have a blank space. Do you get it? So I'm going to sort of label the sequence of events. First of all is that, and then what I'm going to get is I'm going to have like an opened up plasmid right now. See that? Now I'm now going to insert the green gene, that rhymes, into here. I'm going to insert it, and just inserting it is not enough. I need to now kind of tape it in place, get it? Glue it in place. And that gluing process is done by another enzyme called ligase enzyme. So these are all keywords. These are the marks you're going to get. You have to talk about the plasmid, and then the restriction enzyme, and then the ligase. These are the 3 marks basically. And once you have that ligase, you now have this, and this is the recombinant vector. Get it? Because you recombined it with the green gene. So those are where the marks are coming from. So part B, it says the graph shows the effect of temperature on the rate of photosynthesis of tomato plants at 2 different light intensities, high and low. Explain the effect of temperature on the rate of photosynthesis of the tomato plants at high and low light intensities. Okay, so another graph question. Let's be very systematic. The first thing we talk about when we look at the graph is the overall trend, right? And the overall trend is a positive relationship. And tell me how, right? It means that as you increase the temperature, what happens to the rate of photosynthesis? It increases, rate of pho. Okay, I'll write the full word, rate of photosynthesis. That is the first point. Now, the fact that it says explain means that you better think of that second mark here and don't just, move on, is why does higher temperature lead to higher rate of photosynthesis? Because of, you would know this, right? Increased kinetic energy, and therefore what? More frequent collision of what? Collision of enzyme and substrate, right? Enzyme and substrate, because it's the enzyme and the substrate that's doing all of this photosynthesis. More frequent collisions. Okay, so kinetic energy. Temperature, you should always mention kinetic energy. Now, what else do we need to discuss here is the fact that why is the low light intensity lower than the high light intensity? Even though temperature is also increasing the same for both of them. Why would this be lower? So you need to explain that. So that's quite obvious, right? You would say that low light intensity limits photosynthesis, right? Limits photosynthesis. And that is why it only increases up to this far. Do you see that? After that, it becomes a plateau. So limits photosynthesis. But this calls into the question, why, even when you've increased the light intensity to high, it still flattens out. So how do you explain this bit flattening out, huh? How? Number 4, what is another limiting factor then? This goes back to your core knowledge, is the 3 factors that affecting photosynthesis. What are they? Number 1, temperature. You see that here. Number 2, light intensity. You see that here, light intensity. And number 3 is what? It's not on the graph. So you either know it or you don't. It is CO2 concentration. Please know that, guys, because when they ask a question of photosynthesis, they're going to give you 2 and they expect you to know the third one, because that is the 4th point over here, which is at high light intensity. What is the limiting factor now? Tell me. CO2 concentration is a limiting factor. You guys know that limiting factor is a key word for a lot of these photosynthesis, rate of photosynthesis questions. So automatically think of all 3 in your mind as a checklist. The fact that CO2 isn't even shown on the graph, but there's something very suspicious going on here, because even though you've increased the light intensity and the temperature is still plateaus, tells you that CO2 is the limiting factor. Part C, the diagram shows a sustainable glasshouse system that is used in the United Kingdom to grow tomato plants throughout the year. The glasshouse has artificial lighting and heating powered by electricity from a wood-burning power station. So this is power station that is burning wood, and when you burn wood, that is combustion. What are the products of combustion? Carbon dioxide and water. The carbon dioxide is going this way, which then very helpfully supplies the carbon dioxide needed for photosynthesis for the plants, right? And then the water comes up there, and that's the water vapor that's released. Now, carbon dioxide is pumped from the power station into the glass house right there. So this is the setup. And in the question, which is on the next page, but I brought it up here, it says, discuss the advantages of growing tomato plants in this glass house system. In your answer, include the benefits for farmers and for the environment. So we're thinking about the advantages for the farmer and the environment. Now, again, 5 marks, guys. What did I teach you? Having a systematic approach means that before you even start, you think about what kind of stuff, what areas are you going to cover, so that you don't run out of ideas, and even under the time pressure, you don't panic, because you've already laid your sort of a framework for yourself. So just have a think right now. Have a think. Imagine you're the farmer, okay? What matters to you? What are the advantages that the farmer would be very happy about? As a farmer, I would be concerned with 2 things, really. The yield, how much yield am I getting? In this case, the growth of tomatoes, because that's how much I'm going to make money, because I'm going to sell those, right? And the second thing is what? The second thing is usually cost. So I want yield to be high, and I want cost to be low. Like that, okay? Now, what about the environment? What is the environment concerned with? The environment would be concerned with things like, lower pollution, that kind of stuff, right? Lower pollution, and things like, are things renewable or not? Is it renewable energy or not? Is this making sense, guys? This is what I mean by a systematic approach. So I already have 4 overall areas that I'm going to be talking about as I proceed. So let's go into some more depth about this then. Now, I'm going to look at this diagram a bit more, because just now, the question above, we talked about the 3 factors that affect photosynthesis, which is light, and I can see that here. Light, which is here, and carbon dioxide, which is here, And what was the third one? Light, carbon dioxide, and temperature, which is the heater, right? So actually, there's all three conditions required for photosynthesis is already here. So I don't need to like buy any of this. I don't need to buy carbon dioxide or lighting or heater. It's all here. So that's something worth mentioning, right? So let me write this down. So first thing is light, CO2, and heating all supplied. So therefore, there should be no limiting factors. No limiting factors for photosynthesis. So you have everything you need to have some beautiful photosynthesis happening here. And therefore, this should ensure high yield. So just the fact that you have all the stuff that you need means that there should already be high yield or fast production. And just imagine if you're the farmer, right? Like if you don't have enough lighting, then you'd be frustrated because there'll be no photosynthesis. Or if your heater's not working, or there's not enough temperature, or the temperature is not warm enough for optimal temperature, then you'd be frustrated, right? But the fact that they have all three, all perfectly supplied here already shows you that this is going to be very good yield. So you're already addressing the yield part of the farmer bit. Okay, so we've done that already. And then we talk about the cost. For the cost, low cost. Why is this low cost? Because I don't need to buy it. I don't need to buy any. Don't need to buy CO2. Why? Because it's already supplied by this bit here, which is the burning, the wood burning that produces CO2, right? No need to buy CO2. So again, cost saving. So these are the cost aspects. Making sense so far? Okay. Some more consideration is about growing the plants in a glass house as opposed to, for example, growing it out in the open fields. In a glass house, it's all clean, it's all very well maintained, so there's no pests, right? No pests. And that also keeps costs down because you don't need to, buy pesticides to kill off the pests. So there's no pests, bad weather or cold weather or anything of that. Cold weather, because it's all in a very controlled environment in the glass house. Yeah. And then now I'm going to talk about some of the environmental aspects. So what is beneficial from an environmental point of view? I'm using wood burning here in order to supply the CO2. Now, wood is a renewable energy. It's very good to put that kind of word down for anything thinking about the environment. So wood is a renewable energy. So therefore, I'm never going to run out of that. It's a renewable energy. And speaking of that, you can always mention what is not renewable, which is what? Better than fossil fuels, because those are totally non-renewable, right? So it's better than fossil fuels. Just put that key word in, basically, okay? Fossil fuels. Okay, or you can say less use of fossil fuels, which is always a good thing. That's always encouraged in the age of sustainable energy. And then think a little deeper. Look at the diagram, just stare at it, okay? Look at it. The CO2 is directly sort of recycled. Any CO2 produced from the burning of the wood is recycled and goes directly into the glass house, and it goes into the plants for photosynthesis. Therefore, no CO2 is released into the environment. That's a good thing. Why? Why is it good to not have CO2 released into the environment? CO2 not released because it causes global warming, right? CO2 not released to atmosphere. And therefore, there's less greenhouse effect or climate change. You know that, right? CO2 is one of the greenhouse gases. Is there anything else, right? Less ice cap melting and all that kind of stuff, because this is what the environment is concerned with, less ice caps melting. So by sort of organizing, you can see how the first 5 points is about the advantages to the farmer, and then the remaining points is the advantages to the environment. So have a systematic approach and cover different points. Question 10. The photograph shows a scarecrow in a crop field. Let's zoom in and look at the scarecrow. Not very scary at all. Okay, so what does it say? It says scarecrows are models of humans placed into fields to stop birds eating the crops because they're supposed to scare the birds away, right? Design an investigation. Ooh, I'm getting excited when I read this because I love a 6-mark question to tell me to design an investigation. I hope you do too, especially after we go through this. So design an investigation to find out if changing the color of the clothes, so changing the color of the clothes of the scarecrow is wearing alters how effective the scarecrow is at stopping birds eating the crops. Now, why am I highlighting? Because I'm trying to figure out the independent and the dependent variable. So the color, that is the independent variable. Independent variable. That's the clothes, the different colors I'm going to use. And then the dependent variable, that is how effective is it able to stop the birds eating the crop. So this is the dependent variable. So as I'm reading the question, I'm already decoding it. And it says your answer should include experimental details and be written in full sentences. Full sentences. Okay, so guys, I'm sure you know the approach, right? This is a Corms question. So let's have all those things out. C-O-R. And then there's M1, M2, S1, S2. Okay, let's write these down. This is change, organism, repeat, and average, please. Measure again, to 2 things you're measuring, and then same and same. And let's just kind of do a bit more of highlighting. Color coding is one of my favorite things to do because it helps me to understand what's going on. So the change here, this is the independent variable. The dependent variable, that is what you're measuring. That is one of the things that you're measuring, the dependent variable. And then the controlled variables, these are the CV. This stands for control variables. Control variables. So that's what's happening right here. Okay. So now we're able to kind of go through a bit more detail. So for the change, I'm going to be using different colors. Different colors. Color cloth, and you can give more details, the cloths and so on. Now the organism in this case is, well, the scarecrow. And then you can talk a bit about what you're going to keep the same about the scarecrow now that we're here, right? So in fact, this is one of the control variables, but helps you not to forget things. So what are you going to keep the same for the scarecrow? You can keep the size the same. You can use, the same fabric. You don't want to use different materials, right? You're just using different colors. The same size. That this is good enough for now. Okay, so that's organism. And then you're going to repeat, but I'm going to come back to repeat later. I'm first of all talking about the next one, the measure, the dependent variable. So how are you going to measure? What exactly are you measuring? You're trying to think if it's able to stop the birds from eating the crops, right? So you're going to measure the number of birds. Count number of birds. Number of birds. Or what else can you measure, guys? It's not just the birds, it's how the birds are eating the crops, right? So you can also measure the mass of the crops. Because if the birds are coming but they're not eating the crops, then that's not so bad, right? But usually they come to eat the crops. So you should really be measuring the mass of the crops or the plants that are being eaten. So notice I have some unit of measurement. Is the number of birds or the mass of the crops. So remember, you're a science student. You have to give me these details. Now, the second measure is always, for your exam, is always time. Time means in a fixed stated period of time. Think about it, guys. If you're using red, blue, green color for the scarecrow, right? You want to be measuring the number of birds or the crop mass for red cloth for the same amount of time as the blue cloth for the same amount of time as the green cloth. Get it? You can't be measuring, the number of birds for 1 hour when he's wearing red and then just 10 minutes when he's wearing blue. That's not fair. So do you understand the importance of keeping the time the same? And you have to state a period. So what is a suitable period of time? Maybe 1 day, right? 1 day for each color. Understand? So for each of the color of the scarecrow, you're going to measure the number of birds in 1 day. So it's got to be a realistic period of time. And then what are you going to keep the same? this should be quite easy. So the time of the year, because the birds won't be the same, right? So you have to make sure the time of the year or the season is kept the same. What about the weather? If it's bad weather, the bird's not going to come anyways, not because they're scared of the scarecrows, because they don't want to be in the rain. So the weather's got to be the same, windiness, just think everything. Think as much as you can. Temperature is almost always a control factor, because temperature affects many things. Humidity as well, lots of these kind of things, right? Okay. There's much more I can think about. If you're thinking about the crops, you also want to keep the soil pH the same. Lots of stuff. Okay. And then finally, the repeat. What are you repeating? I'm surprised some of my students don't know what they're repeating. So what are you actually repeating? You are repeating the counting of the birds for each of the different colors. And ideally, for each of the colors, you should be repeating 3 times. So red color, repeat 3 times, and then average. Blue color, 3 times average. Green color, 3 times average. Understand? So repeat and then find the average for each color, for each of the different colors. So that's how you do this. There's no way you cannot get full marks on a quorums question if you follow this structure. So another 1-page recap. The purple box here, this is just a heart. This is the anatomy of the heart. This is the atrium, the ventricles, and then you have the pulmonary arteries, the aorta, and the vena cava. But more importantly, these thin arteries on the surface of the heart, these are called the coronary arteries. They're very important because if they get blocked, and they're easy to get blocked because they're so thin, right, then someone would have a heart attack. So that leads down to the red box below. The left-hand side, that's a normal person's artery, and this is someone that's got a very unhealthy diet. And you can see that the artery is clogged up by usually things like plaque and cholesterol. So that will remind you to eat a healthy diet and do regular exercise. Now, the green box compares arteries and veins, the structure of arteries and veins. There's 3 things to compare: the lumen size, the thickness of the wall, and what's inside the wall, and also the presence or absence of valves. So for example, the artery has a small lumen, a thick muscular wall with elastic fibers, and it does not have any valves. Orange box up here, this is just a graph. And remember, if you're ever asked to talk about graphs, you always talk about the general trend, which in this case is inverse relationship, and also any important turning points. And you can see that it kind of stays stable before 5, which is over here, but then it drops. So always think about the overall trend and any turning point, and then try to explain the reasoning behind that. Okay, so pink box here, the pink box here is a typical question on evolution. Remember the main points to mention: mutation, which gives rise to variation in the population and therefore survival advantage. And those that have a survival advantage will survive, reproduce, and pass on the genes. Always say those words. You will get full marks for these evolution or natural selection questions. Now, the yellow box is a genetic question. Remember the difference between genotype and phenotype. Genotype is what's in purple over here. That's the 2 alleles that you inherit, one from your father and one from your mother. So there's always 2 letters there. And then phenotype is what you actually see. So for example, the black ones here, this is the ones with the disease. So that is the phenotype. Remember, this is called a pedigree diagram. And from that, you can figure out the chances of 2 parents having children with or without the disease. Okay, now the blue box here, this is another graph. There's a lot of graph interpretation in biology, so get comfortable. Remember, for this graph, I'm just going to zoom in. Remember, the 3 main points I've put in 3 different colors. The pink color is always talk about the overall trend, which is going up. That's positive relationship between the X and the Y. Then you talk about the green stuff, which is, comparing the fewest and the highest, which is, the fewest risk factor alleles and the highest risk factor alleles. And then the last one is the blue color, which is the lowest risk and the highest risk. So always just find some overall pattern to explain graphs so that you don't kind of miss anything or forget anything, okay? And then for the purple box here, very common question is the question on plasmids and vectors. Remember plasmid, like that, and then the 2 enzymes, which is restriction enzyme and ligase. Put these 2 down and you're going to get most of the marks for that question
if not all. And lastly, the black box is a quorums question. There's always a quorums question. Remember, change, organism, repeat, measure 1, measure 2, and then 2 control variables.
Follow the link in the description box below to download annotated past papers and more resources.